picture this scenario. Two patients with diabetes walk into a clinic. They're like the exact same age. And on paper, their lab results look completely identical.
Right. Same blood sugar, same blood pressure, all that.
Exactly. The same basic metabolic panels. But over the next year, their paths just wildly diverge. Patient A stays completely stable, totally fine. Patient B, though, rapidly progresses to end-stage kidney failure, like suddenly needing dialysis just to survive.
Wow, yeah.
Why the drastic difference? I mean, it's the exact same disease, yet they have entirely different destinies.
It is honestly one of the most frustrating, heartbreaking mysteries in medicine.
Yeah.
Historically, we've kind of liked to think of diabetic kidney disease, or DKD, as this single predictable pathway.
Like a basic wear and tear condition.
Right. But the reality is that it's incredibly heterogeneous. It's not a single path at all. It's this tangled web of different biological battles happening at the microscopic level, deep inside the tissue.
Okay, well, let's unpack this. Because today, we are doing a deep dive into a groundbreaking 2026 study that finally, untangles that web. We're looking at a spatial atlas of the human kidney mapped at an unprecedented single-cell level.
It really is unprecedented.
Yeah. So our mission today is to understand exactly how this new Google Earth-style mapping technology works, discover the hidden immune structures driving that rapid kidney failure in patients like our patient B, and see how this incredibly complex 3D map was distilled down into a simple predictive blood test.
Sounds like a solid plan.
So if you're listening to this right now, I want you to just imagine you are exploring a microscopic three-dimensional landscape of your own body.
And to really appreciate this new map, we kind of need to understand the territory we're dealing with first. So DKD is the leading cause of chronic kidney disease worldwide.
Right.
It affects roughly 40% of all patients with diabetes. Usually, your doctor tracks its progression using a metric called eGFR, or estimated glomerular filtration rate.
Okay, so if you think of your kidney as a highly complex water filtration plant, your GFR is basically just the flow rate, right? Like how efficiently is the fluid moving through the pipes?
That's a great way to visualize it, yes. They also look for albuminuria, which is protein leaking into the urine. That's a sign that the physical filter is damaged. And occasionally they check circulating markers in your blood, like TNF receptors.
But I'm guessing those standard markers have some pretty severe limitations, right? I mean, if they can't tell patient A from patient B, they're systemic.
Exactly.
It feels like they tell you the house is on fire because they smell smoke, but they don't tell you which room the fire started in or, what kind of accelerant is being used.
Yeah, that is precisely the problem. Traditional markers are just an average of your whole system. They don't reveal the actual cell-to-cell biological warfare happening inside the tissue itself. And that's where this study fundamentally changes the game.
By using that new mapping tech.
Right. They use a technology called single-cell spatial transcriptomics. Specifically, platforms like Xenium and CosMx, integrated with single-nucleus RNA sequencing.
Okay, single-cell spatial transcriptomics. That is quite a mouthful. Let's break that down for everyone, because I know traditional RNA sequencing has been around for a while. Usually, when scientists want to know what genes are turned on in a tissue, they just grind up a sample, sequence the RNA, and look at the results.
Yeah, that's the standard bulk or single-cell sequencing.
I love the analogy researchers use for this. Traditional sequencing is basically like making a smoothie out of a fruit basket.
It is exactly like that. You can taste the smoothie and say, all right, I detect strawberries, bananas, and a hint of blueberry.
Right, you know exactly what ingredients were in the basket.
But the physical structure is completely destroyed. You have no idea if the strawberry was sitting next to the banana, or if the blueberries were all clustered together at the bottom.
Yeah, and in an organ as complex as the kidney, location is everything. So how does this new spatial technology fix the smoothie problem?
Well, spatial transcriptomics is like taking a high-resolution, three-dimensional photograph of that intact fruit basket before you blend it. It preserves the 3D architecture.
Oh, wow. So you see everything in place.
Exactly. Through a complex series of chemical tags and high-powered imaging, researchers can look at a highly detailed slice of tissue and see every single cell. More importantly, they attach specific RNA data to precise physical coordinates.
So you don't just know that a gene is turned on, you know exactly which cell turned it on and where that cell sits relative to its neighbor.
Yes, and the scale here is staggering. In this specific study, they mapped over 5 million individual cells across 64 human kidney samples.
Wait, hold on. 5 million individual cells. My brain hurts just trying to visualize that. How do you even make sense of 5 million distinct data points without just getting totally lost? It sounds like an absolute nightmare to analyze.
Oh, it would be impossible for a human brain alone. It requires immense computational power. They use deep generative machine learning models to integrate all these data points.
So the computer does the heavy lifting.
Right. Instead of just listing cells, the algorithms look for complex mathematical patterns, essentially building a dynamic GPS system for the kidney.
A GPS for your organs. I love that. Let's do a quick recap so we don't lose anyone. We've learned that DKD is a wildly unpredictable disease, and this new spatial transcriptomics tech allows for single-cell spatial mapping of the kidney, giving us that intact fruit basket view instead of the smoothie.
Exactly.
So a question for you listening at home. If you could map any organ in your body cell by cell with this Google Earth technology, what hidden neighborhoods would you look for? Because the concept of neighborhoods is exactly how these researchers solve the problem of getting lost in all that data, right?
It is. They mathematically organized the human kidney into 11 distinct niches.
Niches. Okay, if we stick with our city map analogy, niches are kind of like the major boroughs of a city, like Brooklyn or Manhattan.
Yes, perfect analogy.
They're larger multicellular structures that perform major functions. Things like the glomerular niche, where the initial filtering happens, or the vascular niche for blood supply, or the distal convoluted tubule.
Right, and within those large boroughs, you have microenvironments, or MEs. If a niche is a borough, a microenvironment is the specific, localized context of a single street, or even a single apartment building.
Okay, so it's super localized.
Very localized. It's defined by a single cell and its immediate, directly interacting neighbors.
That makes a lot of sense. So when we zoom in on these individual streets, what happens to them when the kidney gets sick? Like, what does diabetic kidney disease actually look like on the map?
Well, as the disease worsens, a specific dangerous neighborhood begins to expand and take over. The researchers identified what they called an injured tubular microenvironment.
Injured tubular, so the tubes are taking damage.
Exactly. The zone is centered around specific cells, IPT and ITIL cells that have become damaged, under the sheer mechanical and chemical stress of filtering thick, sugary blood year after year. These cells basically burn out.
Oh, man.
They stop doing their normal jobs of processing waste, and they start screaming for help.
And what does a cellular scream actually look like? Because I know the paper talks about this creating a profibrotic zone, basically a scarring neighborhood. Does the scream actually cause the scarring?
It does. And because of the spatial technology, we can literally eavesdrop on the molecular chatter between these cells. We aren't just guessing. We can see the physical chemical signals one cell is sending to another.
Eavesdropping on cellular phone calls? That's wild. So what are they actually saying to each other?
Well, in a healthy kidney, the chatter is very stabilizing. You have cells sending signals that correlate with good kidney function, like NRG1, ERBB3, or CXCL9, DPP4 signaling.
Okay, so those are the good guys, the neighborhood watch.
Right, keeping the environment calm. But when things get severe in DKD, the chatter turns toxic. Specifically, the researchers saw a massive uptick in plenar, PLAR, and IL-6, IL-6R signaling.
IL-6. That's a pretty well-known inflammation marker, isn't it? How does it cause damage here?
Let's look at the exact mechanism. This IL-6 chatter was happening heavily between endothelial cells, the cells lining the microscopic blood vessels, and podocytes.
Podocytes, those are the filtering cells, right?
Exactly. They are these incredible specialized cells with little finger-like projections that wrap around the kidney's capillaries. They are the actual physical filter keeping protein out of your urine.
Okay, got it.
So when the endothelial cell gets stressed by the diabetic environment, it releases this IL-6 protein. The IL-6 acts like a blaring fire alarm. It hits the receptor on the podocyte, and the podocyte just panics.
Oh no, what does it do?
It retracts its little fingers, detaches from the vessel wall, and lets protein leak right through.
Wow, so the alarm itself is what breaks the filter.
It's worse than that. That same inflammatory alarm also recruits fibroblasts.
Fibroblasts?
Yeah, cells that act like biological construction workers. They rush to the site of the alarm and start pouring down collagen, basically biological concrete.
Oh, so that's the scarring.
Exactly. This lays down permanent scar tissue, permanently destroying that microenvironment.
That is incredibly vivid. So just to synthesize this subtopic for everyone, we've learned the kidney is spatially organized down to the street level into niches and microenvironments. And in DKD, the stress of the disease causes local cells to broadcast inflammatory alarms like IL-6.
Right.
This panic causes the filtering cells to detach and calls in workers to pave over the area with permanent scar tissue. It's just this localized, destructive feedback loop.
It really is.
So think about your own personal microenvironment for a second. How much do your immediate neighbors, the people closest to you, and their daily chatter influence your state of mind? Because at the cellular level, bad neighbors can literally destroy the block.
And that concept of local influence is actually the perfect segue into the most profound, frankly, shocking discovery of this entire study.
Okay, here's where it gets really interesting. We've talked about the mechanical stress and the scarring, but what about the immune system? Normally, if I understand kidney biology correctly, the immune cells that naturally live in the kidney are pretty peaceful, right?
Yeah, normally they are. They're tissue-resident macrophages.
Right. I picture them like local park rangers, just cleaning up cellular debris, removing dead cells, and generally keeping the peace.
That's how it's supposed to work. But the researchers found that in a very specific subset of patients, our patient B, from the very beginning of the show, that peace is entirely shattered.
What happens?
In these patients, there's a massive, highly organized invasion of B cells and plasma cells.
Wait, B cells? But B cells are the heavy artillery of the immune system. They're the ones that produce antibodies to fight off viruses and bacteria. Why on earth are they invading a diabetic kidney?
Exactly.
I mean, diabetes is a metabolic plumbing issue. Too much sugar damaging the pipes. You're telling me it suddenly turns into an immune system attack.
It is a complete paradigm shift. And what's wild is that these B cells aren't just floating around randomly in the tissue. They physically organize themselves into heavily fortified camps.
Fortified camps.
Yes, they're called tertiary lymphoid structures, or TLS-like structures.
So they are building rogue military bases inside the kidney.
Precisely. If you looked at this under a microscope without knowing the patient's history, it looks exactly like what you would see in a severe autoimmune disease like lupus, or doing the aggressive rejection of a transplanted organ.
But this is happening inside a diabetic kidney. That is crazy. We need to wrestle with this because this is a massive leap. Why would a metabolic disease cause an autoimmune reaction? How does high blood sugar trigger the body to build a military fortress against itself?
It really comes down to chronic tissue damage. As the metabolic stress of diabetes kills off local kidney cells over the years, those cells burst open and release internal debris.
Like proteins and molecules that are normally hidden safely inside the cell.
Exactly. The immune system is constantly patrolling, right? And when it suddenly sees all this strange, exposed internal debris, it misidentifies it as a dangerous foreign invader. It thinks there's a localized infection or a tumor.
Oh, wow. So it gets confused.
Right. So the tissue sends out chemical homing beacons.
Homing beacons? What kind of beacons?
The spatial data map this beautifully. The damaged tissue broadcasts specific chemical trails, specifically CXCL13, CXCR5, and BAFAF-BAFR signaling.
Okay, so these trails seep into the bloodstream.
Exactly. Circulating B cells sniff out these chemical trails, follow them back into the kidney, and use them as blueprints to set up their tertiary lymphoid structures right in the middle of the tissue.
That is just mind-blowing. And they found actively dividing B cells in there, right?
Yes, Ki67 positive B cells expressing genes like AICDA and IRF4. They are actively multiplying and setting up shop.
Unbelievable. But let's get down to the actual damage. Are these fortresses just taking up valuable real estate, or are they actively waging war? Like, how do we know they are the reason patient B's kidney is failing so fast?
Oh, they are actively firing weapons. And to prove this, the researchers had to catch the weapons in the act. They use a technique called IgA pull-down mass spectrometry.
Okay, translation time for those of us who don't run a lab. I imagine mass spectrometry is kind of like a highly sophisticated molecular fishing rod, right?
That's a perfect way to picture it. They cast this molecular fishing rod into the spatial tissue samples to specifically catch IgA antibodies.
Which are the primary weapons produced by B cells.
Exactly. And when they reeled the fishing rod in and analyzed the catch, they found that those antibodies were physically bound to granzymes.
Granzymes? What are those?
Granzymes are essentially packets of cellular poison. They also found them bound to complement proteins, which are like the immune system's smart bombs.
Wait, so the antibodies aren't just like tagging things, they're carrying actual toxic payloads.
Yes. They proved that these rogue B cells are manufacturing localized antibodies, loading them with cellular poison, and dropping these smart bombs directly onto the healthy kidney tissue.
Oh my God.
It accelerates the destruction exponentially. And this discovery completely splits the world of DKD patients into two distinct groups.
The B negative and the B positive.
Right. You had the B negative patients who don't have these fortresses and follow a much slower standard path of scarring. And then you have the B positive patients who are fighting an invisible internal war that destroys the kidney at terrifying speed.
Wow. Okay, so to recap this insane discovery, we found that a specific subset of DKD patients inadvertently trigger rogue, heavily armed B-cell fortresses, those tertiary lymphoid structures, right in their kidneys.
Driven by those chemical homing beacons.
Right, and these fortresses drop local antibodies, basically toxic smart bombs that cause rapid tissue damage. So imagine your own body building a rogue, unauthorized military fortress in the exact place it's supposed to be delicately filtering your waste. How would you dismantle it?
And that question brings us to the ultimate clinical challenge of this entire study. Because while having this beautiful, high-definition 3D spatial map is an incredible scientific achievement, practically speaking, it presents a massive translational problem.
Right, I see the issue. You cannot routinely take surgical biopsies of every single diabetic patient's kidney just to check and see if they have a hidden B-cell fortress.
Exactly. A kidney biopsy is invasive, it carries a risk of bleeding, and it's expensive.
So if we can't physically look inside the kidney of every single patient who walks into the clinic, how do we find the B-positive patients? How do we know who has the fortress?
This is where the researchers had to pull off a brilliant computational leap. They needed to find an echo of that localized tissue damage, something that had leaked out and was circulating in the peripheral blood.
Okay, an echo. I like that.
So they took that massive, complex spatial gene signature, the entire chemical fingerprint of the fortress, and fed it into a machine learning algorithm, specifically an elastic net logistic regression model.
Okay, another algorithm. I'm going to need a plain English breakdown of what an elastic net model actually does. Because it sounds a bit like magic to say you go from a 3D kidney map to a routine blood draw.
Think of it like a smart filter. The algorithm sifts through thousands and thousands of different proteins floating in the patient's blood plasma.
Okay.
Its job is to shrink down the useless noise, the proteins that have nothing to do with the kidney, and catch only the specific combination of proteins that perfectly correlates with the presence of a B-cell fortress in the tissue. But there was a major hurdle here.
What was the hurdle?
The B-positive patients, the ones with the fortresses crashing rapidly, are actually relatively rare compared to the millions of stable diabetic patients out there.
Oh, I see.
If you just feed raw data into a computer, it tends to ignore the rare outliers and just optimize for the average majority. So the researchers had to use specialized data balancing techniques, specifically a method called SMO. Yes. They essentially forced the computer to pay equal attention to the rare, fast-progressing patients as it did the common, stable ones. They had to teach the algorithm how to find a very specific needle in a massive haystack.
So this carefully balanced smart filter does its job. What did it finally spit out?
It successfully distilled those millions of spatial data points down to a highly specific 14 plasma protein panel.
Just 14 proteins.
Just 14 proteins that you can measure with a simple blood test. They call it the B-positive DKD protein set.
That is incredibly elegant. But I have to push back a little here. A computer model is just a theoretical model until you prove it works on living, breathing humans, right? How do we know this 14-protein echo actually predicts kidney failure in the real world?
The clinical validation they performed was incredibly rigorous. They didn't just stop at the lab. First, they tested this 14-protein blood panel on a specialized group of patients called the Trident cohort.
How many people is that?
That was 248 patients. And it worked perfectly. It successfully identified the rapid progressors. But phase 2 was the ultimate test of reality. They took the panel and externally validated it in the UK Biobank, looking at a massive external cohort of 3,309 participants with diabetes.
Wow. Over 3,000 patients. Okay, that is real-world population-level proof. What were the actual results? Like, did it beat the standard tests doctors are currently using?
It significantly outperformed the current clinical gold standard, which is a tool called the kidney failure risk equation. In statistical terms, the new protein panel boosted the predictive accuracy, something called the area under the curve, or AUC, from 0.63 to 0.70.
To put that AUC jump in perspective for anyone not steeped in statistics, think of it like grading a test. A 63 is a solid D. A 70 gets you to a C-.
Right.
Now, that might not sound like an A+, but in the incredibly muddy, unpredictable waters of predictive medicine, jumping from a 63 to a 70 across a population of thousands is a staggering improvement. It means catching thousands of patients who would have otherwise fallen through the cracks.
Exactly. The study showed that patients who scored in the top quartile of this new protein test progressed to complete kidney failure drastically faster than the rest of the group.
Okay, so what does this all mean? We have a blood test. We find the B-positive patients. Then what do? If we know it's a rogue B-cell base camp causing the destruction, can't we just attack the B cells? We must have drugs for that, right?
We absolutely do. B-cell targeted therapies, drugs that specifically deplete or inhibit B cells, have existed for years. They're widely used right now to treat autoimmune diseases like lupus, rheumatoid arthritis, and even certain types of blood cancers.
But they've never been used for diabetic kidney disease.
Because until this microscopic map was built, nobody knew that B cells were the primary culprits in this specific subset of DKD patients.
Wow.
Now, armed with just a simple blood test, a doctor can screen a diabetic patient, identify if they have the B-positive signature, and potentially enroll them in clinical trials to receive existing FDA-approved therapies that wipe out those immune fortresses before they destroy the organ. It is the very definition of precision medicine.
So we are looking at a future where a routine blood draw could entirely replace a surgical biopsy, guiding a perfectly tailored, life-saving treatment plan. It's astonishing. Let's do a final quick recap of this blood test translation.
Sure.
Machine learning basically translated that huge spatial map into a simple 14-protein blood test, and they validated it in over 3,000 patients, proving it predicts kidney failure better than current clinical models.
Yes, beautifully summarized.
So ask yourself, could a simple blood test one day entirely replace a surgical biopsy for you, guiding a perfectly tailored treatment plan?
It's looking very likely.
Let's pull all of this together. We started this deep dive looking at a mind-bogglingly complex 5 million cell spatial map of the human kidney. We saw how this Google Earth view revealed the localized chatter of injured cells screaming for help, creating dense pockets of scarring.
The microenvironments.
Right, and hidden within that scarring, we uncovered a shocking secret. Rogue, highly structured B-cell fortresses acting like an aggressive autoimmune disease, dropping toxic smart bombs inside a diabetic kidney.
It really is wild.
But most importantly, this wasn't just an exercise in mapping for the sake of mapping. The researchers successfully distilled that entire 3D fortress into a 14-protein blood test that can literally predict the future of a patient's kidney and open the door to immediate targeted treatments.
It is a phenomenal example of how mapping disease at the fundamental cellular level translates directly into actionable, life-altering clinical tools. But if we zoom out for a second, this discovery raises an incredibly profound, almost unsettling biological question.
Oh, I have a feeling I know where you're going with this.
For decades, we've classified diabetes strictly as a metabolic disease, right? Wear and tear. High blood sugar and blood pressure damaging the pikes.
Yeah, purely metabolic.
But if a purely metabolic stressor like diabetes can somehow trigger our immune systems to form these highly organized, rogue autoimmune fortresses in the kidney, where else in the body might this exact same process be happening right now?
Oh, wow. That is a chilling thought.
Right. Could other chronic, supposedly metabolic conditions like heart disease, atherosclerosis, or even neurodegenerative diseases like Alzheimer's actually be hiding these localized autoimmune battles?
Wait, really? You think there could be fortresses there too?
Are there microscopic B-cell fortresses secretly hidden in the plaques of our arteries or deep in the tissues of our brains that we simply haven't mapped yet?
It makes you completely rethink how we categorize disease altogether. Patient A and patient B. On paper, their metabolic markers looked exactly the same. But now we finally have the map to see the invisible war raging inside patient B. The X-ray machine for the microscopic world is finally turned on. So keep questioning the map, because as we've learned today, the territory is always much deeper and much more complex than it first appears.